Hi, everybody. I am Sue Tappan. I'm a member of the SPARC Data and Resource Center. And today we're going to be talking to John Tompkins as part of our latest installment of Bridging the Body and the Brain seminar series. We're thrilled to have John here to tell us all about the important work he and his team have been working on in terms of the heart's little brain, the ICN, and we're going to hear all about it in just a few minutes. So John is picking up on his series followed right after Dr. Naveen Jayaprakash, who gave us a talk about the vagus nerve and how it's organized and all of the intricacies that are involved in that are relevant for building neuromodulatory devices and understanding how communication between organs and the brain are passed. And our next speaker in the series will be Dr. Vaughan Macefield from Monash University in Australia, and he'll be bringing us back to the vagus nerve to talk about intraneural microelectrodes and how you can get a better understanding of the intravesicular communication properties in the human vagus nerve. So before we get into that exciting topic, I want to tell you about the SPARC Codathon. So each year we've offered a grand opportunity for people to get together and explore the SPARC portal to develop new ways of interacting with the data, the tools, and the resources on the SPARC portal. And it is available for anybody in any country to participate. We have multiple teams distributed throughout the world. We build those teams with those who do write code, those who are scientists who help provide the biological context of what is being built and how that can be useful for other scientists to take advantage of. And right now we're in the team assignment phase. So you can sign up right now to be a part of this great codathon. The codathon will happen the first weekend in August. It's a 3-day period where you get together, you create something new, and you have a chance to earn actual real live cash. And so I hope that you'll take a moment to scan that QR code, check out what's available, and sign up to participate. It's always a lot of fun. But the whole reason why the Codathon exists and why this seminar series exists is because we are talking about the SPARC portal. The SPARC portal is an open neuroscience and systems physiology platform that allows us to bring together the body and the brain, from organ signaling to peripheral nervous system information to in-depth information about neuromodulatory opportunities to impact organ function. So we have recently been accepted by AWS, Amazon Web Services, into their open data partnership. And so that allows us to be able to offer the data that is available on SPARC to everyone free of charge for storage as well as for egress. So when you contribute your data to the SPARC portal, it will be stored free of charge via AWS. And you can then access your data and anybody else's data of interest from the cloud as well without charge. We are an NIH and an NIH approved repository, and we're also HEAL compliant. So that means that if you have funding from the HEAL initiative, you can choose to have your data hosted on SPARC. And from there, it'll be cross-referenced into the HEAL data ecosystem. And so it allows an even greater opportunity for people to find and discover and utilize the information from the careful research that you're doing. So what can you do with SPARC? As I said, the portal itself is more than just a data repository. Excuse me. We obviously are very interested in being able to have exceptional experimental data, computer simulations, and models available about as research objects available on the portal, but we also create mechanisms to allow you to utilize that data, view it in new ways, and explore it. And so we have anatomical maps where individual research data is assimilated onto a map so you can see the context in which they're presented, as well as a data knowledge management system that allows us to keep track of the connectivity between organs and neuron populations so that you can start to build these circuits of understanding of how these different parts of the autonomic nervous system are integrated together. This data and models can be utilized on our cloud platform called OSPARC. This is free to use, and we invite you to discover it and play with it yourself. This is a grand opportunity for you to create computational pipelines to analyze your own data. You can bring in data that you find on the SPARC portal. You can actually put those two things together and create something entirely new. And those are just a few things that you can do on the SPARC portal. So we invite everyone to become part of the community. We invite you to contribute your data, your models, your resources to the community so that all of us can work closer, faster, and more integrated together to create new solutions to biological problems. The way you can do that is in two ways. You can contribute individual sets of data. We call those individual submissions. They receive professional curation. So one of the things that sets the SPARC portal apart is that all data sets undergo intense curation to ensure that they're harmonized, aligned, and utilize a structured format. We allow you through that process, these data sets receive a DOI, which can be reserved so that you can include that DOI in the manuscript that you're writing to interpret the data that you are contributing to SPARC. It can be held in embargo until such time as your paper is published so that everything becomes available at the same time. This allows you to build a bigger picture, a bigger view of your body of work, because it allows you to not only see the interpretations and the ways that you're putting the data from an individual experiment into context with other published observations, but it also becomes a citable object itself. So it is added to your ORCID profile. You can include it in your CV. SPARC datasets are multimodal, so we accept data across species, across different experimental paradigms. We also accept code and simulations as well. So it's really meant to be a place for your entire experiment so that you're not putting your data up into individual silos. Additionally, we encourage you to consider writing SPARC as your repository of choice when you're submitting grants. This allows you to indicate to your funding organization that you are committed to fair and open science, and all of the great curation and support that SPARC offers is available to you at that time too. You can scan the QR code there and learn more about that and get right in touch with us if you'd like to take advantage of these particular things. on the SPARC portal, we have over 600 contributors who have provided over data that encompasses over 11,000 subjects, 11,000 samples, 4,000 subjects, 54 tissues, and 7 species, getting right to that multimodal aspect that I was just talking about. All of those individual components of subjects and species are organized into over 237 research data sets, 46 anatomical models, and 46 computational models. This number is growing every month, and in fact is up by just one, as we have John's data set available as well. All of these are available 100% for your reuse. So it's we're really looking to build a community that can help us go further faster. Data sets of all different types are accessed and utilized. So every research data sets have been accessed and downloaded nearly 8,000 times. And the computational models and anatomical models are also heavily accessed as well, indicating that the effort that you undergo to make your data fair results in access. People are interested in what you do and how you do it. And so with that, just to bring this all back full circle, we have initiated this seminar series as a way to introduce everybody to just the broad scope of areas of expertise and aspects of knowledge that are being contributed by researchers to the SPARC portal. And today we have Dr. John Tompkins. He's from UCLA, and he'll be talking about the little brain and the heart and the differences in the intrinsic cardiac ganglion neurons between species. So John, welcome, and thanks for being here.
Thank you, Sue, and thank you for your leadership of this SPARC seminar series. It's a real asset to be able to have these talks available. Let me share my screen and start my presentation. So I've got about a 40 to 45 minute talk for you this morning or afternoon, whatever time zone you might be calling in from. I want to try to save time for questions at the end, so I'll try to move through this at a pretty good clip. I think we're going to hold questions till the end, which Sue will moderate, but I hope you will chime in, and I'm happy to try and answer those. So as she mentioned, today is in part kind of a celebration for us, because I'm happy to say that this data set is live as of this morning on the SPARC portal, and I'm going to show you today how you can find that. And I'm going to kind of be giving you a tour about, with respects to my data set, what exactly you're going to find when you go there. And I'm going to be talking about this project, which has been SPARC funded. And this was kind of the initial phase of SPARC, doing the basic science. And as I'm a cellular neurophysiologist working with the group here at UCLA to study these intrinsic cardiac neurons that reside on the heart and make up what we like to call, the little brain of the heart. And so on the background here, I wanted to show you that you're looking here at a slice. So this was a slice of a human intracardiac ganglion isolated from the right atrial ganglionated plexus. And this was done in preparation for doing a transmission electron microscopy, but we've isolated the whole ganglion, fixed it formaldehyde, glutaraldehyde, embedded it in an epoxy resin, and then did thin sections. And this is a light photomicrograph where we just assess tissue health after sectioning, and then we take it to the electron microscope. I'll be sharing those EM images later in the talk, but I just think this is a beautiful image to show you kind of the complexity of these ganglia. So these large cells, sorry there's no scale bar, but they're about 40 microns in diameter. These are intracardiac neurons, the neurons that reside on your heart on its epicardial surface. You see nuclei here of satellite glial cells that surround them. And then within this neuropil, I mean, this looks like any neural tissue you've seen from the brain or spinal cord, that we're seeing cross sections of axons and dendrites. And it's just to show you the complexity of these little miniature brains, these ganglia, which reside on the heart. This is the paper that goes with the data set. It was published first of this year in the Journal of Physiology, and find it there. You'll find it as part of this special issue of the Journal of Physiology on cardiac neurobiology concepts to clinic. And there's a lot of beautiful work in this issue. Also, importantly, there's a special memoriam to my boss, colleague, Jeff Ardell, who passed away the first part of this year. But he, many of you are familiar with his work. He's kind of contributed, was a mainstay, a godfather in this area of understanding the intrinsic cardiac nervous system. So what does this little brain of the heart do? So on the left side, I want to just show you, this is a really simplified diagram of cardiac neural control. And it's showing kind of the extrinsic nerves, right? The heart is innervated by autonomic nerves, both the motor nerves carrying efferent neurotransmission to the heart, as well as afferent nerves, which are sensing the interstitial milieu within the heart to relay and modulate that motor activity. And while commands are coming from higher centers of the neural axis, they can be modulated locally within the heart's intrinsic cardiac nervous system, which is this network made up of both these extrinsic nerve fibers as well as the intrinsic neurons, such as the intrinsic cardiac ganglia. So the autonomic efferents, we have a sympathetic innervation, and it's a two-cell pathway to the heart from the spinal cord for sympathetics. Primary neurons project to sympathetic ganglia, for example, in the paravertebral chain, synapse onto a postganglionic neuron, which then, so we have a short projection, and then the second cell has a long projection to the heart. In contrast, the parasympathetic neurons, the principals or first cells reside in the brainstem. They project all the way to the heart, where they're going to synapse within these intrinsic cardiac ganglia onto the postganglionic neurons, which are residing on the epicardial surface. Now, what do these cells do? Well, they modulate a number of components of cardiac function. They innervate the SA node, they innervate the atrial muscle, the AV node, ventricular muscle, as well as the coronary vasculature. And so this is all going to affect the chronotropy, or the timing of the heart rate, dromotropy, conduction from the atria to the ventricle, inotropy, the contractility of the myocardium, as well as the lusitropy, or the relaxation of the myocardium after contraction. All these things are regulating cardiac output in a way to be able to meet physiologic demands under changing stressful conditions, and are certainly important in affecting our health and well-being to maintain normal cardiac control. Now, an analogy that I kind of like to use, if you're familiar with automotive mechanics, is that in many ways, this is like the engine control unit on an internal combustion engine, which all modern cars have. And this is the computer system that controls engine function. It controls spark timing. It controls the mixture of fuel and air by feedback from all of these sensors throughout the system, which are feeding in, and then ultimately getting control from our brains through the gas pedal to go or the brake pedal to stop. And as far as the control of the heart, the gas is like the sympathetics, and the brake are these parasympathetics. And so to better understand this system, particularly across species, we set out to develop a structural and functional atlas of these intracardiac neurons across in mice, in pigs, and importantly in human tissues. And this is one of the first studies to be able to do this in human tissue. I'd like to note that one of my colleagues, Joanna Montgomery, has another beautiful paper in that same issue of Journal of Physiology, and where they too are presenting some of the first data from these human intracardiac neurons. But we've looked at the neurophysiology of these cells with sharp electrodes. We've backfilled them to look at single cell morphologies. I'm going to show you some of the histology within the ganglia as well as the ultrastructure. But importantly, if you look up this paper, you're going to find in the data statement at the bottom of the paper, you'll find this link, which this has been a broken link, but I'm happy to say now that if we click on this, see if this will pop up, it's going to take you directly to the sparc.science website, the SPARC portal, and this is the data set. And so we've got about 1,300 files here. There's about 1 terabyte of data. And I'm going to be showing you now, I'm not going to go through this website now because this is just live, but you can find all of this data that I'm going to be talking about at that site by following that link from the publication. Okay, so why these 3 species? Obviously, these are commonly used laboratory research animals. We all use mice because of the ability for genomic manipulation. Pigs are more common as a preclinical model, and we're getting into a large mammal model, which is closer in some of its physiology to humans. But ultimately, the goal of most of our work is to affect human disease and to ease patient suffering, improve quality of life. So, and what I'm going to share today is that there are important differences. I mean, we've diverged from these species, eons ago, and pigs are actually more closely related to aquatic mammals. And of course, our family primates are our closest relatives. But I'm going to show you kind of how different these cells can be between these species. All right, I want to just acknowledge all of my collaborators, the people that have generated the data. So in my lab, I've been helped by Janaki and Youngjan, and we isolate whole ganglia, do the sharp electrode recordings and 3D morphology. Don Hoover is a colleague at East Tennessee State University who did the immunohistochemistry with a tech, Liz Smith, in his lab. And Leif Hafton was the one overseeing the TEM work, and his postdoc at the time, Natalia Biscolia, was the one really spending all the time in the scope. Importantly, I want to acknowledge the contributions by One Legacy. They're the organ donor provider within the Los Angeles area. Dr. Ajajola was instrumental in facilitating our access and availability to that tissue. Dr. Mori, although not an author on the manuscript, was often there with me. So typically we were receiving these hearts in the middle of the night to begin experiments at 1 or 2 a.m. On these isolated cells. And of course, I want to acknowledge the leadership of Dr. Shiv Kumar at the Cardiac Arrhythmia Center, as well as Jeff Ardell for their helpful discussions and input on this work along the way. Okay, so to begin, so where are these epicardial ganglia? Well, they're distributed across the surface of the heart. And this wasn't really a goal of the paper. There are a number of outstanding publications that I would direct your attention to, particularly this work by Danius Pauza, where he's looked at whole heart patterns of innervation using a cholinesterase stain. It's beautiful work to kind of show you the full distribution of nerve fibers and ganglia. Also, I want to note this a recent paper by Jack Chung's group, South Florida, for the mouse heart. They've done really beautiful confocal imaging. But this is the first figure from our publication, and it's important here just to show kind of the region and the ganglia that I'm focused on for this study. that most all the ganglia that we've isolated are from an area that we would call the right atrial ganglionated plexus. So in mice here, you see a whole mount preparation of mouse heart, right, left atria. And most of the ganglia on the mouse heart are situated around the pulmonary veins, which you can see here in this cartoon. And this one that's close to the SVC was something that I could always go to and find a ganglia. And these are multiple examples of these ganglia. And this is what I would isolate from mouse. In pig, we have a more circumscribed area. Peter Hanna led a really nice study on this. You can find in Circulation Research, using multimodality to look at these RAGP neurons. But in the mini pig in particular, it's a beautiful model because the location of these ganglia are very well demarcated by the presence of these epicardial fat pads. And we're here on the dorsal surface of the atrium between the SVC and IVC. There's an RAGP fat pad. And beneath that, if you lift that up, you'll find these ganglionated plexi, meaning that they're these ganglia, which are these clusters of neurons distributed in this web-like network of nerve fibers. In the human heart, here you can see the whole human heart. This is the location of the RAGP that's on our hearts. And this is an example of one of those ganglionated plexi isolated from humans. And this just shows an image that, and I want you to appreciate that while these are the areas we focused on, that these GPs, ganglionated plexi, are distributed around specific regions of the heart. And these drawings by Drew Armour, Rene Cardinal, show for the canine heart some of these major ganglia, like the RAGP, or the inferior vena cava, inferior left atria, which is on top of the coronary sinus. And there's also ganglia which are located with preferential innervation of the ventricle. So in the database, one thing you'll find are these 3D confocal scans of these ganglia isolated from the different hearts. And so, you know, this isn't something you get from a publication. Of course, in the publication, you just see the 2D images, but I'll play through these. And I want you to appreciate that, you know, these are 3D scans, Z-stack images that you can scroll through, and you can actually reslice them in multiple dimensions to get an idea of the total thickness, how many cell layers, how many cells in a ganglia. And I'll just point something out to you. One, you should notice here the difference in scale bars. So the mouse cells are much smaller, about half the size of those from pig or human. And the mouse neurons are also densely compacted. They're tightly, very close together. And in the human's ganglia, we tend to see that there's more space. And as I'm going to show you as we go through some of the nerve fills and look at TEM images, that this is because these cells largely lack dendrites, and the synapses are really going to be on the cell body, whereas in the pig and human, pig's kind of in between the mouse and human. But particularly in human, there's a complex dendritic arbor around these cells, which is where, and so has to be space for that within the ganglia. Next, I'm going to show you some of Don Hoover's findings regarding the immunohistochemical characterization, or the neurochemical coding. So what do we find as far as principal neurotransmitters, neuropeptides in the ganglia? So based on the vesicular acetylcholine transferase staining, so this is a protein that packs vesicles with acetylcholine. And we use it as a marker for cholinergic neurons. And what you see is this kind of punctate labeling around cells, which is marking where these vesicles, which are filled with acetylcholine. So that kind of identifies the preganglionic parasympathetic inputs to these cells. But also when you look at the cell bodies themselves, they're clearly making these proteins. And we believe that most of these cells are what we would call the postganglionic parasympathetic neurons controlling the heart. Not all of them, though, and we'll say that we also find tyrosine hydroxylase, which is a marker for sympathetic neurons, but it's one we have to be a little bit cautious of. And this is because, you know, TH is pretty far upstream in the synthesis of catecholamines. I mean, it's really saying that these are dopaminergic or can make dopamine, but they have to have the dopamine beta hydroxylase and other downstream enzymes to be able to make norepinephrine, and then also the vesicular monoamine transporter to actually package it and release it. And in some early studies by Don and Eberhard Way also did a beautiful paper showing that the mouse cells lack all those other proteins. And so that while these may be TH positive, we can't say for certain that they are functionally adrenergic. However, in humans, they do find those other markers. They are DBH positive, they are VMAT positive. And so we think that in larger mammals, and especially humans, that there may be more of a mix of both postganglionic parasympathetic as well as some sympathetic neurons. Now, the human cells are also unique in the fact that they were the only species that we see. So these are TH positive fibers encircling a human intrinsic cardiac neuron, and that punctate labeling of those synaptic boutons really suggests that is likely an interaction of sympathetic nerve terminals onto these cells. So that is not something, Jack Chang, again, in his mouse study, did a beautiful study of it in mouse, and they'd never identified these, and we haven't identified them, and we don't identify them in pig either. Importantly, here to the right side, I give kind of what is our perception after looking at many images of the relative abundance of these TH positive cells or fibers. And I'm going to show a cartoon in a little bit that will give a kind of an overall summary of the presence of these neuropeptides. There is some nNOS in there as well, but it was largely absent from the human tissue. So what you see here that looks like staining is actually autofluorescence from the tissue itself. These are lipofuscin granules that we can clearly see with EM, and which autofluoresce and have this pattern. So we don't think those are nNOS positive cells, but we do see positive cells in pig and mouse. Couple of other neuropeptides of interest. NPY is present in many of these neurons within the mouse intracardiac ganglia. It's also present in some of the pig cells. So this is likely a peptide co-transmitter with these cells, which are either TH or adrenergic or most likely cholinergic. And then in human, we see kind of a dense network of NPY positive immunoreactive fibers, some of these which are co-localizing with tyrosine hydroxylase and may be of extrinsic source, as we see fewer NPY positive cells within the human ganglia. Vasoactive intestinal polypeptide, part of the secretin glucagon superfamily, PACAP relative, is also there, but it's almost totally absent in pig, but is markedly abundant in human ganglia. And so certainly as far as understanding the physiologic role of these ganglia and neurotransmitters used, I think VIP is going to prove to be something important to study in human. These are sensory markers of sensory fibers, substance P, calcitonin gene-related peptide. We also see co-localized with one another, abundant in mouse ganglia, relatively sparse in the pig. Surprisingly, we do see some substance P or CGRP positive fibers in pig, but not near the abundance of what we see in mouse or in human. Human, there's also a dense meshwork. And so this cartoon I put together is just kind of one way to visualize these species differences in the neurochemical coding of both the neurons and the nerve fibers. So the nerve fibers, you know, unless we do kind of an explant study, we can't say for certain whether they are of extrinsic origin, meaning they have their cell bodies outside the heart, or whether they're intrinsic. And here the darker labeled species mean they have a greater abundance, and lighter labeled. And it just kind of gives a picture that that there's a lot of variability between species, particularly in the expression patterns of these neuropeptides. We next really need to look at their functional roles. So next, I'm going to talk a little bit about the single cell morphology of these cells. So here are these representative ganglia from each species. And so while I'm recording, I'll talk about the electrophysiology of these cells next, but we backfill them with neurobiotin and then fix and stain with a secondary streptavidin conjugated secondary. And then on the confocal, again, do 3D scans, and then we can develop these structures using software from Microbrightfield. This is using Neurolucidor 360 to create these tracings. And again, what you'll find in the database is that you can actually download the 3D versions. You can download, or Microbrightfield has provided an app for you to be able to zoom or scroll these in 3D, but I mean, you can even 3D print these. There are a number of things that you can do with these structures, and I'll talk about that in a couple of slides. But what I want to show is just the differences here across species. So these are nerves, intrinsic cardiac neurons from the mouse, and what you'll see is that they're pretty simple. So we filled 14. I think we targeted about 30. We get about a 50% success rate with this approach. But this is the cell body, and they all just have a single axon, which doesn't really appear to branch, at least not near the cell body. And then rarely do we ever see the presence of dendrites, but they tend to be short and stay close to the cell body. In contrast, in pigs, we begin to see, larger mammal. These cells are about twice the size of the mouse cells, and we see now these more complex dendritic arbors. As well, we see that some of the axons appear to have bifurcations. We don't know if these are functional collaterals of these axons, but we do find more branching. And here's another evidence here in this cell where the blue axon branches, usually close to the cell body. In the human neurons now, we go up in even an order of complexity as far as dendritic complexity. This is kind of shown by the amount of dendrite length here close to the cell body. In the paper, you'll see this figure where we've kind of quantified all of these different parameters, numbers of stems, bifurcations, and branches, and we just see kind of this increasing complexity of the dendritic arbor. One thing to note, so, mice here are about 10 to 14 weeks of age, which they're juveniles. The pigs that are about 6 to 8 months, which are also juveniles. The human tissue that we're getting they're typically 40 to 60 years of age. And so we don't know how much age might also affect the growth or development of those dendrites. So another thing that these can be used for is to begin to build computer models to be able to simulate function. So I'm in the process of uploading these structures to neuromorpho.org. You can also get them from the SPARC portal. And then if you're familiar with programs like Neuron, you can then put that structure into Neuron, where we then can map ion conductances to different segments or compartments of the neuron to begin to develop models of how these cells are functioning. I'll draw your attention to another paper that I was happy to be a part of that was led by Raj Vadapalli and Jim Schwaber at TJU, and as well as Bill Litton, kind of the godfather of this neuron tracing and creating software-based models of neuron function. And here they used a lot of the data from the RAGP and in combination with their transcriptomics approach to look at ion channel expression and present models for different types of neuron firing. So that's in that same issue, and I recommend that you check that out. But this is a goal of our work and a direction that we're heading to kind of better understand this intrinsic cardiac nervous system. So next, I'm going to show some of the EM data, and then I'll wrap up by talking about the electrophysiology. So here we're looking at this a mouse intrinsic cardiac neuron. So this is the cell body, and the arrows are indicating synaptic boutons. And particularly, I'll show this one in the box. The boxed areas are shown at the bottom. You can see this image in the paper. And what you can clearly see in that these synapses are on the cell body. And so here we have the pre and post synaptic densities. You can see the small clear vesicles and the presence of a mitochondria in here, a couple of mitochondria. Now, in contrast, when we look at pigs, ICNs, and we did 3 animals, about 30 cells were sampled. We never see axosomatic synapses. So if you look around the margins of the cell body, you won't find them. You have to look out next to the cell body and to find these examples where this synapse is being formed with a dendrite. And so again, the postsynaptic density, lots of mitochondria. And then you can see within that synaptic bouton, these small clear vesicles. They're also evidence for large dense core vesicles, and these are what are likely containing the neuropeptides. And humans as well never see axosomatic connections. They're only axodendritic. And again, we can see small clear vesicles, synaptic specialization. And one thing I was particularly struck by is that we had such clear images of these small clear vesicles that we measured their diameters in at least at 3 different terminals, but we had more than 50 vesicles to measure. And I was quite surprised to find that the pig and human vesicles are almost twice the size of the ones in mouse. And I would have thought that something that is so important to quantal content of these neurotransmitters that are being released could be, I would think that would be evolutionarily conserved, but whatever the regulatory elements are that affect the size of those vesicles appear to be different in humans. And I don't know that literature well enough to know what is known, but I think that that's something also interesting to follow up on. These are kind of cartoon depictions of these neurons between the 3 species. This relatively simple neuron from the mouse receiving these axosomatic synapses. And I'm going to show you the functional recordings next of kind of the implication of this, these being the pig and human, where we have dendritic arbors, and all those connections are going to be out on the dendrites, not on the cell bodies. And so to functionally study these, as I said, we've done sharp electrode recordings from individual neurons, and then I use extracellular stimulating electrodes placed on intraganglionic nerves to look at synaptically evoked potentials. And so this figure shows you examples of these synaptically evoked potentials. So here panel A is from mouse, B from pig, and C from human. And in mouse, what we see, so my schematic kind of illustrates, we know we have an axosomatic connection, and when you stimulate that preganglionic input, we're resting here at about minus 50 millivolts. We get a really large excitatory postsynaptic potential, which is always super threshold for a spike. And if we stimulate with trains of stimuli with different interspike intervals correlating to 5, 10, or 20 hertz, they follow perfectly with no failures. And I think that in these animals, that these ganglia are really functioning kind of as that classic relay station. There's no time for a heart going 500-600 beats a minute to be doing a lot of neural integration there on the heart, although there are neuromodulators present that are changing. But now in pig, we see something totally different, that we see a convergence of inputs from different nerves, some of them having this strong phenotype that often give you a spike, and some of them weak, which almost never give a spike. And when we give intertrained stimuli, they follow very poorly. And so it appears that a convergence of input is really necessary. Otherwise, these cells are kind of functioning as a low pass filter, that they're not allowing high frequency input to pass through. Now, the human cells are a little bit like mouse and pig, in that we see a convergence of inputs of both strong and weak, but they tend to have a high following frequency. And that's because of the difference in the excitability of these neurons, which I'm going to talk about in a couple of slides. last thing, I won't belabor this, but I want to talk about that we've measured the latencies of these nerve inputs. So latency is the measure of time from the stimulation until we first begin to see the depolarization. And so this is a measure of conduction down the axon and time for vesicular mobilization release crossing the synaptic cleft. And this latency values in mice are tightly clustered to about 2.5, 3 milliseconds. They're significantly longer, around 5 milliseconds in pigs, but we see something totally different in human cells, which I'll show here. So these are the short latencies of mouse, longer latencies of pig, but now in human, we see very long latencies. And I don't yet know exactly how to explain this. I don't know if it could be an artifact of the preparation, or in this example where we're seeing nerve evoked potentials 38 and 91 milliseconds after our 100 microsecond stimulation pulse, it really suggests to me that there are likely multisynaptic pathways connecting. And I know there's some evidence for the idea of interneurons within these ganglia. And this is something else that we're working to follow up on to find out how much these cells are kind of communicating with one another. As far as basic membrane physiology goes, the spikes look pretty similar between species for the most part. The mouse cells rested at a little bit less negative membrane potential. This could be because the cells are just much smaller, even though I use a very high resistance, 120, 130 megohm sharp electrode. There could be damage to those, but the spike profiles, AP duration, half widths, they look about the same. But one thing that did strike us as being very different are the after hyperpolarizations. And in particular, the human neurons have these whopping huge AHPs. And I'm going to show you in the next couple of slides, part of what we know is are the conductances that are playing a role there. Again, all of the ePhys data is going to be available for you to download and look at through the SPARC portal. So that was kind of one clue that the expression of ion channels between these species is different. And now we've only done transcriptomics in pigs. We've not yet done it in humans. We're working towards that. But when I'm doing these experiments, I also measure the excitability of the cells by giving. So these are current clamp recordings. We're recording membrane potential. We inject current 100 picoamp steps in a graded fashion. And cells that only fire a single spike, we call them phasic. This is classic nomenclature. And then those which can fire multiple spikes repeatedly and usually increasing numbers with greater depolarization are tonic. And in mice, we find about 2/3 phasic and about 1/3 of them are tonically firing. And this pertains to how excitable the neuronal membrane is to synaptic inputs and its ability to fire repeatedly. In pigs, we almost never see tonic neurons, 100%, at least in the 40 or so cells sampled in this study. Now, I did have to call Jim and Raj at TJU because they had predicted from transcriptomics that there would be tonic neurons. And after about 60 neurons, I did start to find a couple of tonic cells. So this data set is a bit incomplete in that regard. There can be tonic cells as they predicted. So it's a strength of the transcriptomics. They're just very rare. But in contrast, the human cells, they're all tonic. And so the ion channels and their activation properties and inactivation properties are apparently unique between species. These are some of the currents that regulate excitability, H current, M current. In the next couple of slides, I'm going to show you the involvement of A current and this small calcium-activated potassium conductance known as the SK current. So to inhibit SK, we use apimin. This comes from bee venom. Nature makes the best medicine. This has nanomolar potency, highly selective for these SK channels. And so in mice and humans, we see that it greatly shortens the duration of the after hyperpolarization. And so that means that this calcium influx during the spike and activation of these small conductance K channels is what is in part mediating that AHP. And when we do that in mice, I mean, it makes them go nuts that they start from being phasic and then they go to tonically firing and they're reaching, you know, 60 spikes a second. It's a much higher frequency that we can ever see in pig or human. Interestingly, in pig, apimin didn't really do anything to the AHP, but it did change excitability. And for the cells that are almost always phasic or only give you a single spike, it can turn them into tonic neurons. 4AP now to inhibit A current, which is a potassium conductance involved in the repolarization of the action potential. It performs as expected in mouse and human, that it broadens the half width of the AP. So we've blocked that repolarizing conductance. But again, here in pig, it had almost no effect on the duration of the AHP. We didn't see a change in excitability with 4AP in mouse or human, but there was a small change in excitability in pig, despite the lack of that effect. So there are clearly differences here in what ion channels are functioning on these neurons between these different species. So I'll wrap up then, try to save a little time for questions. So, you know, what we've tried to do here, we've created a database of these structural and functional properties of intracardiac neurons from these 3 species. And we identified here from this descriptive study these kind of characteristics of evolutionary specialization of these neurons in each species, which is likely related to the different ways in which their hearts work. Obviously, when we went from walking around on 4 legs to standing up and walking on 2, this put new hemodynamic load affecting preload and afterload on the heart. And so we need a different neural network to be able to compensate those changes, to be able to maintain my cardiac output. Highlight this, that a lot more work needs to be done to better understand, particularly what types of neuronal cell types we have. And I think, you know, transcriptomics is probably going to be the next step for doing this. But as we've seen done for the stellate ganglia and more recently for dorsal root ganglion neurons, identifying, you know, specific functional classes of neurons. So is a neuron that goes to the SA node different than one that's projecting to the ventricular myocardium? My guess is yes, but we don't yet know that. Lastly, you know, just a request to all of you that are working in this area or any area of autonomic neuroscience, you know, help support this open data initiative. Share your data, especially the human data, because it's so rare, so difficult to come by. And let's build more comprehensive data sets to work towards building these cellular models that you guys can compete with on the SPARC Codathon that can really aid. Our ultimate goal is to develop neuromodulatory approaches for cardiac disease, because we know that these cells are affected. They contribute to atrial and ventricular arrhythmias. They can be responsible for sudden cardiac death. They're involved in atrial fibrillation. They're involved in vasovagal syncope. And so being able to target selectively this neural network, we can alleviate patient suffering, improve quality of life. Lastly, just a shout out to everyone in the SPARC DRC that has making this data available, especially to Sue, as I've said, that we've corresponded quite a bit in getting this talk together, and also special shout out to Anka Pilko. She's helped me. So anyone that's tried to publish a data set, I want you to know it's like publishing another manuscript. There's a lot of work involved, but all of these people make it a lot easier. So I'll just conclude here. Let me thank all of you for your attention. And I want to leave you with this quote from Santiago Ramon y Cajal. So, you know, his work was really beautiful stuff, always chokes me up. But of course, he used a sparse labeling technique like we're using, but he was using Golgi stain. But he said, you know, the cortex in the brain it's like a garden filled with innumerable trees, right? These neurons look like trees. That's why we call them dendritic arbors. And the unceasing activity of the brain as a whole is like the hum of a restless beehive, right? That hum are the neurotransmitters, right, acting on the biophysical properties of these cells. And I hope you realize from today that there's a similar complexity within this little brain of the heart. And we have to understand the morphology of the plants in that garden and what are the neurotransmitters that are so important there. So please visit the SPARC portal to learn more, and I hope you enjoy the data. Thank you.
Thank you, John. That was
Yes.
incredible. I loved the journey you took us on. I invite everybody to pop your questions in the location there on the Zoom panel to start a conversation while we've got John right here for us. John, I'm going to ask a question that I've been wondering about since you started showing these differential differences between the different species. I wanted to ask about the model system of the pig. It looks so different from the human and seems to behave so much functionally differently to the different applications that you applied. How do you choose between mouse or pig when you're looking at modeling human heart function?
Well, I think there are a lot of problems with the mouse, particularly if you're trying to do arrhythmia studies. You know, just the mass effect, a much smaller heart. You know, they're tuned to go 400 to 600 beats a minute. It's just very different. You know, the pig heart, like the human heart, it thumps along at about 70 beats a minute. It's similar in mass and size, and you can do arrhythmia science on those hearts. But you know, pigs are expensive, and a lot of people don't have the resources. So there's still obviously a lot of valuable work that can be done in the mouse. Um, I just think, you know, you really have to think about what type of question you're asking and choose the model species, you know, most appropriate for those answers. And that's one thing I hope that this data set shares is that, you know, if you're trying to look at integration of neural inputs on intrinsic cardiac neurons, mouse is not the model you want to use. Rat is a little better, and you know, that's something I hope that maybe we can add is, you know, I've spent a lot of time also using guinea pigs. I don't know much about what the morphology of those cells, but you know, finding the most appropriate models is, again, it's going to be related to the question you're asking, and
All right, I got a couple more to throw at you. Are you ready?
Yes, please.
Okay, so Julia would like to know if you've seen any different ePhys properties, if they change with any kind of cardiopathology like fibrillation. You seen that?
Great question. So I would refer her really to Joanna Montgomery's study. So Joanna's focus that's in that same issue, they took neurons from patients undergoing a coronary artery bypass, and they separated them into two patient pools, ones that had AFib and one that did not. And they saw that the spike frequency was different. It was shorter. They all had tonic neurons, but the interspike intervals were shorter in patients with atrial fibrillation. So our tissue is largely, it's a mixed bag. We do have in the portal, in the manifest, we've got kind of all the EKG and some of the medical diagnostics as far as what the pathology was. Most hearts we're using have been rejected for transplant, so there's something wrong with them. But we did get a couple of healthy patients. They were 40 years old, they were in an injury, and the hearts were otherwise normal. But I think the disease is it's a complex question, and we don't have a data set with enough numbers to stratify. But as far as the tonic-like excitability, we found even in healthy patients, they're all hyperexcitable, but not enough different disease groups to answer that question.
Do you see any trip channels in those neurons?
We didn't stain for trip channels. I'm sure that they're there on the sensory nerves. We know they're there for the sensory nerves.
Mhm.
Um, but no, it's not been something I've stained for. And I don't know about the efferent neurons, the parasympathetic neurons, whether they have trip channels. But it's certainly something worth looking into.
Okay. So another question from another very avid listener wants to know a bit more about the parasympathetic as compared to sympathetic postganglionic neurons in the ICNs. So do you see a difference in number? And do you think the ratio changes between para and sympathetic between the different species? Any thoughts on that?
Well, yes, kind of, what I alluded to. So in mouse in particularly, like I said, even though we see TH staining, don't assume that those are sympathetic neurons. Um, we really think in mouse, as in guinea pig, so Rod Parsons, my old boss, has done some beautiful work. He really thought that all the neurons in the guinea pig are postganglionic parasympathetic neurons. Now, I know Jeff's work in canine models, they really find evidence for both sympathetic and parasympathetic. So there is variability between species. When you're just looking at immunohistochemical markers, you've got to include a full panel like the DBH, vesicular monoamine transferase, to know if they're sympathetic. I don't know what this TH expression means. We do see that it changes with disease, and I don't know if these neurons, have some ability to change neurotransmitter under stress. But definitely in the human tissue, uh, there is clear evidence that there is a population of adrenergic or sympathetic neurons within the ICN that are residing on the heart, and there are these synaptic terminals on the parasympathetic neurons. So that kind of crosstalk between sympathetic and parasympathetic is greater in humans than in the mouse.
Excellent answer. That's very compelling. If anybody has any other questions, please pop them in. I have one more that I'd like to ask you to get your perspective. So you did explain that the subject's age was quite different between the different species. And so if we focus only on the larger mammals, you did use juvenile pigs, but most of the subjects that the human subjects were adults. And I know that pigs get large as they age. So do you have any concept or thoughts on whether or not you would expect the dendritic structure of, the morphology of these cells to be different in a younger human versus an older human, or an older pig versus what you've seen for the younger pig? Like how do you have
Yeah,
any
so
idea of what you think the maturity of those arbors when that hits?
I I don't know offhand. Something someone can do, or we could even do with the data on the SPARC portal, because we've given the age of all the subjects that data comes from, and it's all cross-linked. So you can do simple correlation plot to see if there's an increase in complexity or maybe a loss in complexity with age.
Right.
Um, and pigs, in other animals, we know, for example, like excitability of the neurons change with age. Usually they're hyperexcitable when they're younger, and they become less excitable when they age. And we also know that, dendritic arbors, I don't know as much growing in size, but their organization obviously is changing with experience. I will say, that we're using the mini pig as a model, and the reason we've switched to that, we were using the Yorkshire pig, which is like our food stock animal, and those have been bred to just get very large in a very short amount of time, thousands of pounds. But the mini pigs stay small, and we can keep them chronically. We're doing studies now, and this is a study that's going to be forthcoming. I'll put a plug for myself that we're looking at properties of neurons before and after myocardial infarction. And that's done in older animals because we've had to infarct them, keep them around for 8 weeks, and then measure. So we'll be able to do some age-dependent studies in those animals, but we're also looking at a greater number of ganglia, RAGP, IVCILA, as well as the VIV, to see if membrane properties or neurons are different in different ganglia that have different spheres of influence, and what is the impact of myocardial infarction on those cells. So that study is coming, and it'll be out end of the year.
That's awesome. Is that something you'll be presenting at SFN this year?
I'm not presenting that at SFN, but I am presenting a work I've done with Dr. Ajijola looking at changes in the excitability of stellate ganglion neurons in mice after MI.
Okay, cool. I'm going to close with this last question, and I'm bringing this one up because it gets right back to your roots, which is physiology. We've hit you with a lot of questions that aren't specifically physiology based, so I figure we've got to give you one that you can knock out of the park. Not that you haven't done that for all the other questions. I just mean that it hits your sweet spot. So the question is, do you have any experiments planned where you stimulate the sympathetic and parasympathetic preganglionic neurons and then analyze the combined effect on the postganglionic? Any thoughts?
Yes, I'd love to do that. It's a difficult experiment to do. So one thing I will say that most of the time we'll wash on some hexamethonium when we're doing these nerve evoke, which is going to block the ganglion nicotinic receptors, and that knocks out 95% of the nerve evoke responses. So as far as the fast synaptic excitation, it's principally mediated by acetylcholine. So, finding a sympathetic nerve, knowing that it's sympathetic. I mean, one thing we can do is we can puff on or spritz norepinephrine to see if it has an effect. We know it's got presynaptic effects, postsynaptic. But identifying, I mean, we could do it, I guess, with nerve stimulations and then staining afterwards to identify if there were sympathetic inputs to the nerve. But something I haven't alluded to that will also be forthcoming, though, is that I didn't show the slow responses. So often when you give a tetanic train in particular, you can see either a slow depolarization of the membrane potential or a slow hyperpolarization. And so we're still kind of working out. So I think that the slow depolarizations are often due to acetylcholine activation of muscarinic receptors and inhibition of a current known as M current. The hyperpolarizations, I'm not totally certain what those are mediated by. I think that the sympathetics and the release of norepinephrine might have an effect on some of those slow responses. So clearly, more experiments required to delineate the responsiveness to application of like norepinephrine.
Thank you so much. Thank you, John, and thank you everybody for attending. John, we really appreciate you sharing your data on the SPARC portal and giving everyone an opportunity to dive in and see all of the great depth and open opportunities that such a rich data set can provide. And thanks for all of your great work so far. Can't wait to see what comes next.
Thank you. Thank you, everyone, for participating, logging in. Take care.
All right. Take care, everybody. Till next time.